Go back to my
if you just go back.
There.
Good?
Good?
Yeah. All right. So we'll go ahead and get started. I'm Bethany Sargent. I'm the Outreach Coordinator with the Clean Water Initiative Program, and I have Tina Heath and Charlie Hohn, who are Wetlands Ecologists with our Wetlands Program. And they're going to be talking about the Wetlands Bioassessment. For folks online, if you wouldn't mind if you have questions to just type them into the instant message box, and I can field the questions in the room. And then folks in the room obviously can ask questions, and Tina and Charlie would prefer just for clarifying questions during the presentation, and then they'll leave some time at the end if there are questions you have outside of the scope of what they've presented so far for any follow-up questions. So I'll go ahead and turn it over to Tina.
Thanks, Bethany. Yeah, so I'm Tina Heath. I work in the Wetlands Program alongside Charlie Hohn and our newest employee, Levi Casey. And together we make up the Wetlands Bioassessment and Monitoring team. And today, Charlie and I are going to talk to you guys about all things bioassessment, because some of you may not know what we exactly do. But I bet most of you do know, though, that wetlands are critical to our watersheds, and they serve as these interfaces between where land and water meet, and as a result, they provide imperative functions to human health, water quality protection, and wildlife habitat. And so they are essential on the landscape for us in terms of environmental and human health, and in turn, we should be studying the health of these ecosystems in order to provide better insight into how to manage, protect, and restore these important areas, as well as to provide like a more complete picture of overall watershed health. So the outline for today is that I will be going over the program purpose, objectives, and history. I'll be going over some of our current methodologies that we're looking at. I'll talk about a little bit of how we're currently developing biocriteria for wetlands, and then Charlie, I'll shift things over to Charlie, and he'll be talking about some of our data analysis procedures and results, some exciting new projects that we're starting this summer and hopefully moving forward with, and then the future of our program in terms of our goals and what we're intending to do. And then, yeah, we'll provide time at the end for any questions or discussions. So why do we have this program in the first place? So the purpose of the project is to build a pertinent and practical wetland bioassessment program to assess the wetland biological integrity, as well as determine overall ecological condition of Vermont's wetlands. So it is expected that over the long term, the results of this program should help to improve regulatory and permitting decisions, should help provide significant information to rehabilitation and restoration projects in terms of viability and success rates. And then it should help us to identify effects of anthropogenic stressors and environmental stressors on wetlands over the long term. And so we use 3 different types of survey methods to look at wetlands across the state, and these survey methods are basically modeled off of the EPA's approach to wetland monitoring. So we have level 1, level 2, and level 3 type of assessments. And so level 1 assessments are these broad landscape scale assessments. And so this is basically happening like in the office as a desktop review, where we're using ArcGIS, looking at orthophotography, LiDAR, etc., getting some basic information on not only the wetland we want to go to, but also looking at the surrounding land use areas, the intensity of the surrounding land use areas, any sort of stressors that we can identify, just to get an overall sense of what we're about to sample. Level 2 assessments are these rapid assessments at the wetland scale, so it's pretty site specific. And this is where we're going to those wetlands and we're doing relatively quick and simple field protocols to gather basic ecological and physical information on the wetland that we're at. We're using right now what is called the VRAM, the Vermont Rapid Assessment Method, to do that sort of work. And what's important, though, is that we want to make sure that these rapid assessments are validated and calibrated to our level 3 type of assessment work. So level 3 assessments are these site-intensive biological assessments. So we're going to these wetlands and we're collecting biological information, physical information, as well as chemical information. We're collecting a suite of water quality parameters as well. So this is the type of assessment that will really provide insight and information onto the wetlands condition. So how do we do all of this? How do we measure wetland health? Well, so we conduct assessments of wetlands across the condition gradient. So we're looking at all different types of wetlands, along with everything from super hammered disturbed sites all the way up to high quality reference type conditions, and then everything that falls in between on that spectrum. We're gathering chemical and physical data at each of these wetland sites. That includes water quality, hydrology information, soils information, landscape characteristics, as well as any stressors that may be present in or near those wetlands. We're sampling and describing the vegetation at these sites in order to develop vegetation-related metrics of wetland biological integrity. We're completing rapid assessments and evaluating the ability of those rapid assessment methods to reflect the overall wetland condition. And then we're hoping to expand the use of these metrics in assessing the overall ecological health of our wetlands. So this is just a brief history of the program, but what's crazy to me is, after really looking at what we've done, we've been around for almost 20 years now, which is awesome. So the project started in the late 1990s as this EPA-funded pilot project that specifically looked at vernal pools and northern white cedar swamps. And so this project focused on the collection of macroinvertebrates and water quality samples, and then did some condition ranking within those specific types of communities. A final report was produced in 2003 outlining those results. In 2006, the program restarted again and was housed within the Wetlands Program. And at the time, the program really just focused on stream and lake associated wetlands so that the project could capitalize on the expertise of other individuals who did similar monitoring methodologies. And so we used their help to really kind of develop our program. And we also used a condition ranking method known as the Human Disturbance Ranking, the HDR, which was adopted from the late 1990s project. 2008 up to 2011, the program shifted to looking at all different types of wetlands, and we refined our condition ranking methods. So we used the HDR for a couple of years, but then we switched when we created the VRAM, the Vermont Rapid Assessment Method, because it helped to capture more relevant data that we needed in order to get a better all reflective score of condition. And we had to drop macroinvertebrate sampling because we lost funding for it, so we focused on just using flora and water quality. 2011, the program participated in the first ever EPA National Wetland Condition Assessment, known as the NWCA. And it was this nationwide effort among other participating states to collect information on wetlands across the nation using a standardized approach. And we also still conducted state bioassessment work, and it was a really big year for us in terms of work. I remember how busy that field season was. In 2012, the program focused on reference quality wetlands in the sense to look for the potential for sentinel sites, for these long-term monitoring sites. And at this time period, we were introduced to what is called the Floristic Quality Assessment Index, the FQAI. It's a certain vegetation metric that was currently being developed in the Northeast. 2014, we adopted NWCA methods in order to stay more in line with the EPA and have a more standardized approach to our field methodologies and site selection processes. But what was really awesome about those years was that we finally created a database that could house our data and analyze our data and have a functional working database, which was huge for us. 2016 was our second round of NWCA, and the highlight of that year, and maybe for all of these years, was that we were finally able to hire a full-time staff position purely dedicated to bioassessment, not ripped away into regulatory, not a seasonal employee. We had our own dedicated person, and that's Charlie Hohn's position. So that was huge for our program, because up until then, we only had seasonal and temporary staff to be able to carry the program forward through the year. So it was always a start, stop process. And so it was really hard to develop the program. So last year was a huge year for us because we had Charlie as our fearless leader, and so we did a lot of different things, which, we dropped the NWCA methodologies for site selection and field work just because it was too time intensive, too cumbersome, just not enough for our staff's capacity. So what we did was that we changed our field methodologies to be more in line with the natural heritage inventory methodologies so that we could start to share data between the two programs, because they have a ton of historical plant data that we could use. We also classified wetlands at the natural community type scale. We became more integrative with other water quality monitoring programs in terms of prioritized site selection. We refined our vegetation metrics based on some of the results we're starting to find, and we modified and expanded the use of VRAM. So it was a really, really big, great year. So our current approach towards sampling wetlands, and we're hoping to keep moving forward more consistently into the future with, is that we are collecting data on a 5-year rotational basin schedule, meaning that we're concentrating most of our sampling efforts into specific basins that are up for sampling in a given year. And then in 5 years, we will go back to those same set of basins. And so we're more in line now with other monitoring programs as well. So across the division, we now have most of our monitoring programs and their efforts being kind of defined into these certain areas of Vermont. We have a more integrated approach for targeted site selection, meaning that we are now working more closely with our basin planners, looking at their tactical basin plans, looking at their prioritized monitoring needs, in the sense that if they have some class 1 candidate work that they need to be done, we go and we look and assess those wetlands. If there are some potential restoration sites that they would like us to look at, we'll add those to our list. And we also work with the other monitoring program folks in the sense that we're sort of taking advantage of existing data that they have in other lakes and streams and sub-watersheds. And we're going to those areas to see if wetlands are kind of reflecting the same sort of condition score that some of these other media are also reflecting in those sub-watersheds. When we are at our field sites to do level 3 assessment work, this field season usually takes place between mid-June through early September so that we can accurately identify plants in the field. So we're collecting biological, chemical, and physical data. And in terms of biological, we are looking at the vegetation at the site. So we're setting up specific plant plots in representative areas of the wetland, and we're collecting plant information such as identifying plants down to the species level, percent coverage of certain species, as well as strata information. For chemical data, we're doing a suite of water quality parameters. So we're looking at everything from phosphorus, nitrogen, turbidity, chloride, alkalinity, conductivity, pH, you name it, a whole suite of parameters that we've been consistently sampling for over the last 10, 15 years. And then we're also collecting physical data, such as soil and hydrology characterization of the site, and also looking to see if there are any stressors present. Once we're done with those level 3 assessment duties, we then utilize the VRAM and do a rapid assessment. We're collecting additional information about the wetland and the surrounding area and getting a sort of overall quality score for that wetland. Once we're done, once Charlie has time in the office, he goes back and refines the mapping that he did before we went out and may modify boundaries, may change the natural community typing a little bit, maybe identify some stressors that we didn't see out there, etc. And I guess what is important for me to point out is that with last year and now really moving forward is that we're now classifying wetlands using the Vermont Heritage Natural Community methodology. So we're kind of narrowing our scope in terms of grouping wetlands and types of wetlands together. And so that kind of segues into our discussion of wetland biocriteria and where we're currently at in terms of development. And so basically we need a system that can provide a measure of wetland biological integrity. And so we're using the vegetation at these sites as that indicator. And so we're using a vegetation metric known as the Floristic Quality Assessment Index, the FQAI. And so the FQAI was designed to assess the overall naturalness of an area based upon the species that are found in that particular area, and then those species specificity to that particular habitat. So the FQAI basically rates human disturbance by accounting for the presence of native and non-native taxa in a given area that we're looking at. And it does that by looking at what is called the coefficient of conservatism for each plant species. And so this is called a COC score, and it's rated on a scale from 0 to 10. And it basically indicates the tolerance level of a plant species towards disturbance, as well as indicate that particular plant species habitat requirements. So if that all sounds like what the heck am I talking about, I'll try to give you guys a couple of examples to put it in perspective. So the COC scale is a scale from 0 to 10. And so a couple examples. So we have like purple loosestrife, which is an invasive non-native species. So that plant will receive a 0 as a COC score because it's not native. Pretty easy to understand in that sense. And then we go to the other end of the spectrum where we take like pitcher plant, where that will receive a score of 10 because they're only found in very specific places, bogs. And so that plant has a very specific ecological habitat requirement, and we're assuming that it really can't tolerate too much human disturbance or even maybe natural disturbance as well. So it gets a really high score because it's very specific to a certain habitat type and tolerance level. And then there's a lot of species that fall into that spectrum, all in the middle. And another good example is like red maple. Red maple receives a COC score of 2, which sounds like, is red maple like a bad species? No, it's not bad. It's just the fact that it's so common. It's found in both uplands, it's found in wetlands, it's just everywhere. And so it's a generalist species, and that means that it can be found in many different types of habitats, and it can probably tolerate a lot of disturbance both anthropogenically and environmentally. And so we're using the COC basically to really look at the condition of these wetlands. So the FQAI and the COC are great measures of biological condition, but they really only work best when compared to other similar natural community types or wetland types. So in the past, we have been kind of classifying wetlands into broad categories using the Cowardin classification system. So we would put emergent wetlands together, forested wetlands together, shrub wetlands together. But the FQAI and the COC does not really work well when we're trying to compare all of those different types together. So, for example, with forested wetlands, we have like a high-quality northern white cedar swamp, and then we have a high-quality silver maple riverine floodplain forest. And so they're both high-quality wetlands, but they're not going to have the same COC score. One is going to have a higher score than the other. And it's not based on its condition. It's just the fact that they're simply different plant communities that have been influenced by, they're on different parts of the landscape. They have different hydrological regimes. They have different soil pHs, different water chemistry, etc. So, of course, they're going to have different plant species growing there. So to try to compare those two systems together is like comparing apples to oranges. So you really, in order for the FQAI, the COC metrics to work, you really need to be comparing a northern white cedar swamp to another northern white cedar swamp. And so we've been starting to be able to draw connections between the COC, wetland condition, and natural community type. We've started to expand kind of like our focus on not just strict FQAI values, because FQAI values essentially account for species richness at a site. And so when we're looking at the mean COC values of a site, we're starting to find that it relates a little bit better with condition, and that it also alleviates some of the statistical problems that can be associated with the FQAI, because our plant plots aren't always the same size and dimensions. So like, for example, our current methodologies, we use like 10 by 10 meter plots in emergent wetlands and shrub wetlands, but 20 by 20 meter plots in forested wetlands. And then in years prior, we've used all different sorts of plant plot sizes to collect plant information. So looking at like the mean COC score of a wetland site kind of alleviates that species richness factor that can kind of skew the data a little bit, and it creates more of a like level playing field to be able to compare these condition values a little bit easier among similar wetland types. And so, like I said, we are finding these connections between COC, wetland condition, and natural community type. We're also working closely with the natural heritage inventory folks, and we've been able to collect a huge wealth of data, plant data from them. They have over 400 wetland plant plot data for us to use, since they've been collecting plant information for the last 35 years at all of these like amazing reference quality wetlands. So we've been able to use that information, generate COC scores from their plant plots, and start to really develop condition gradients, or the start of condition gradients, for a lot of different types of wetlands based on just the information we've gathered from those guys. So really cool things are happening. But I should warn that although the FQAI is a great measure for condition, it should not be the only measurement for the condition of a site. A lot of wetlands don't fit neatly and nicely into natural community types. We often find a lot of disturbed wetlands, beaver influenced wetlands, where plants are just doing their own thing and just we have no idea what's going on. So that can be problematic at times, and so that's why it's important to not only look at the biological data that we're finding, but also including the physical data, the water chemistry data, to have a better overall representation of what's actually happening at that site. And then lastly, before I shift things over to Charlie, I did want to talk about our Vermont Rapid Assessment Method. That's our level 2 sort of rapid assessment protocol, the VRAM. And it's a really important tool for us, and we've been utilizing it for a long time, and it's got a lot of value for us. So the VRAM was adopted from the ORAM, the Ohio Rapid Assessment Method, a while ago, and it was modified to more accurately reflect Vermont's wetlands. And so it looks at a number of things. So it looks at the condition of our wetland site in terms of looking at measures of buffer intactness, how large the buffer is, the intensity of the surrounding land use, what stressors are present, etc. It looks at the function of that wetland, so it looks at the hydrology of the wetland, the plant structure of the wetland, and the wetland size. It looks at the value of that particular site, so is it a special wetland, meaning is it an uncommon wetland, is it exemplary, is it a really rare wetland type, does it contain RTE species, so that would boost up its value. And then it provides this overall quality aspect of the wetland, which really combines the condition, function, and value together. And so it provides this overall quality score, which is a numeric score that falls on a scale of 0 to 100. And so, like I said, we've been using the VRAM for years now, and we've been utilizing it and expanding the use of it over the last couple of years. So we have a lot of VRAM data to the point where we're actually able to sort of create a condition gradient just based on the VRAM scores. And so what we've been finding is that from 0 to 65 is where a lot of disturbed wetlands lie on that spectrum. From 65 to 85 is where we'll see a lot of moderate quality wetlands, and then 85 to 100 is where we really start to find high quality reference condition wetlands. So it's been a really important tool for us. And what is exciting, and Charlie will start to talk more about this in just a minute or so, but we are finding correlations between the VRAM scores, between the COC scores, between certain water quality parameters, and even natural heritage condition ranking used by the heritage program. And we're starting to find these really cool correlations. So it's kind of nice to see that like the VRAM is validating some of our level 3 assessment work, and that also like the level 3 assessment work is validating our VRAM as well. So lots of exciting things that Charlie will start to talk a little more about.
Awesome.
I'll just switch chairs.
Yeah. Hello, I'm Charlie. Awesome. So, yeah, what are we finding with all of this stuff? We are finding a lot of data, first of all. So one thing I went and did is I went through the data set we have and looked for the most complete individual data sets, meaning I went and looked through for data that has full water quality information, has a plant plot, and the plant plot was in a recognizable either natural community or in some cases a disturbed wetland, but one I could recognize as having a single likely natural community within the plant plot. And that constituted this 100 data points, which we used for some of our core analysis, because then we were able to compare all these different factors together. We also, as of the end of 2017, had 275 VRAMs. I've actually done a couple dozen more since then, but they aren't included in this assessment. But yeah, those are a good example of wetlands all over the state that we visited. Most of those are skewed towards the last 2 years when we've had more time to look at VRAM, but that also includes some going back much longer. And as Tina mentioned, we have over 400 natural community plots that Natural Heritage Inventory has allowed us to use the data for. And these are really wonderful because it's part of their, they assess significant natural communities. It's something that, for instance, Eric Sorensen works on. I actually used to work on that program as well. But so they go and try to find the best example of every wetland type as described in Wetland, Woodland, Wildland, a book some of you may be familiar with. And some types have more samples done in them than others because of how their inventories work. But there's tons of data on cedar swamps, tons of data on bogs, things like that. And so for those wetlands that we have lots of data on, this is giving us an idea of what a really good example of these wetlands looks like. And they also have their own internal condition ranking, so we can use that as well. But this is just allowing us this baseline because, like Tina said, every wetland type is showing up as having a different kind of scoring a little differently. And so now we can look at, oh, wow, well, let's see what, now we have a pool of data to look at how they have scored in the past or now, and then compare it with what we find now, which allows us to target other wetland types and disturbed wetlands and compare it. And there's also a lot of wetland data out there that aren't in our data set yet. So another thing that we've been working on is just collaborating with different people. We've been talking to people at UVM. We've been doing some VRAM training and encouraging other groups to do VRAM because it's easy to learn. We've had Forest Service send us some other watershed management groups, different people, and that'll be expanding. And also, when a wetland permit is issued, a lot of times there's vegetation data associated with that we might be able to use. So over time, we'll be looking at trying to round that up. Other things Natural Heritage Inventory collects. And we're also continuing to work on how we display this data. Right now, there's one watershed data portal, which I'll talk about in a minute, but that's only for the full Tier 3 plots. Well, how can we share VRAM with everyone? How can we share natural community maps more fully, et cetera, et cetera? So that's an ongoing part of our program. And so this right here is the watershed data portal, which has data going back to 2006, even though it didn't exist yet then. But so this is a really cool thing that's visible to all of the Watershed Management Division that gives you a ton of data. I want to get up and point, but then people on Skype can't see it. But basically, you put in the plant vegetation data, and then it spits out all this, and the VRAM, and then it spits out all this information. Some of these we talked about, some of them we haven't. Some of them are corresponding more with other condition indices than others, but you'll see on there there's all kinds of more stuff we can dig into over time, like how many sedges and grasses are in each plot, the number of invasive species found, and a whole bunch of other statistics. We can always add new things to this as they come up too. So basically it's just giving us this deep pool of data that we can then continue to look at for trends over time. And this is, as Tina mentioned, we're also kind of looking at, well, how well do these work to tell us about wetland condition and function? And, there's always lots of caveats because ecology is really complex, but we have, even when you don't consider natural communities, this is just lumping every bit of data we have, you can see that there is definitely a trend where wetlands, if you look at this graph on here, you'll see the VRAM score and the coefficient of conservation where, in theory, if you found something like a bog that literally had only pitcher plant, it would get a 10, but in practice, the highest we've seen is about 8, and likewise, the lowest one we have is about 2, which had a lot of invasive species, but presumably a few natives too. And I'll mention that this is not taking cover into account. You can get a lot lower score if you go into a patch of invasive common reed where the cover of common reed is 95%. You end up basically having the score for common reed for that plot. So, but this one actually correlates a bit more closely with our VRAMs. And one really important caveat is that VRAM is intended for the entire wetland, whereas the plots are only for a 10 by 10 or 20 by 20 meter section. And so this is something we're continuing to look at, like how do we link data of different spatial areas. And one of the things we can do is actually look at, if we have a wetland that gets a VRAM that says poor, medium, or reference, what range of numbers will the plots get within that? So, it's an ongoing look at this data, but in general, you can see, beyond just looking at the trend, you can see this pattern where really, really high scoring wetlands in terms of the coefficient of conservation almost always have a high VRAM score. However, things with a low VRAM score can range a little bit from low to medium in the coefficient of conservation. For instance, if you think about going into a large wetland, like a VRAM could be something like on the scale of, Victory, that whole Victory wetland complex, or the Missisquoi Delta or something. You might go in there and place a plot in a more disturbed area and get a lower rating, and then go and place a plot in a better condition area of the wetland and get a higher rating. So these are all things. But then when you start looking at natural community, it makes it easier to pull that out too, or doing smaller subset VRAMs. But it just gives you an idea that it may make more sense rather than trying to look one-to-one to try to break the VRAMs into groups and look at it that way. And that's something we've also been looking at. You'll see that the one wetland that got an 100 VRAM doesn't even have the highest score because it's this vast beaver wetland in wonderful condition. But as Tina mentioned, a beaver wetland has a lot of natural disturbance, and the COC doesn't necessarily know what causes the disturbance. And a lot of generalist plant, there are actually no invasive species in there, but it was a lot of goldenrod. It was this wonderful pollinator habitat, but it just was one of these things where we need to remember it's a beaver wetland. And then maybe as we start looking at beaver wetlands and as we get more data and split that out, we might say, well, that really did, maybe 4.5 is really good for a beaver wetland. And that's something we're still looking at, and I'll mention that again. But we're also the other thing I've done is I've looked at when we have access to it or when it's been done. The Natural Heritage Inventory has a methodology where they rank a natural community from A through D, whereas D is for destroyed, basically. It's very poor condition. A is basically an old growth forest or a bog with a deep heat mat, something like that. And I didn't want to fill this thing with so many scatter plots that everyone got sick of looking at them, but there is a correlation there as well, we've seen. And then we're being able to start breaking that out by natural community type. Another thing I'll mention is the FQAI is being reviewed, and there's new numbers coming out. We haven't had time to put them into our system or look at whether they correlate more or less than the old numbers, but they're more by bioregion. Like if you think about balsam fir, if you find balsam fir in the Nullhegan Basin, it's everywhere. If you're in, Shoreham and find balsam fir in a wetland, it's probably a very specific habitat that allows it to grow there. So they'll be breaking off the Champlain Valley into a different bioregion, so it'll give different numbers that way, which is going to be really nice. They've also, I think, done a little bit to address like what Tina was talking about, how red maple is such a generalist. Some of those yellow birch would also score really lowly, and they looked at that, and I think they bumped those up a little bit, saying, well, they are generalists, but they also are in a way specialists. Like red maple may be occurring in early successional post-logging forest, but it's also occurring in wetlands with a lot of nutrients where other trees can't grow. So, they're just reevaluating those numbers, and we'll see how that ends up turning out over time. These are going to be hard to read, so don't worry about looking at what everything says, but this is just me showing that there's different natural community types, and the COC they get is very different. Like you see one that's like a super overachiever, and that's open peatlands, bogs, and fens. And one of the interesting thing to think about is, what do you call a disturbed peatland? Basically, you call it a cattail swamp. When a peatland has very severe disturbance, then the peat starts breaking down. And this can even happen from a beaver. This isn't necessarily human disturbance. But when a peatland experiences changes in water level or increased nutrients, a lot of times it stops being a peatland. So if we went into a cattail marsh and 100 years ago it was a peatland, it would be in the pool of cattail marshes, which falls under the one of the other types. And so it's just one of the marshes and sedge meadows. So, this is something we'll be looking at over time too, but it gives you, other than the peatlands are the ones that stand out the most, but you'll also see that wet shores, which is like basically areas along rivers and lakes, score lower. And yeah, those areas do have a lot of human impact, but they're also areas where there's a lot of natural erosion, disturbance, things like that. So it makes sense they would score lower. Floodplain forest is one. In other states, it's been even more pronounced, but like Maine has found this even more so, but they tend to score low because there's, even in a perfectly pristine floodplain forest, because floods come through and generalist species come in. It doesn't mean there's anything wrong with them, but as we gather more and more data, we'll just be able to compare floodplain forests and see. Shrub swamps is an interesting one because it has a very wide range. And I think the reason that is because shrub swamps range from alder swamps in disturbed areas to things like sweetgale swamps that are in a peatland. So that may be more a function of how shrub swamps are classified, and we may end up breaking those out into a smaller subgrouping. I started looking at this, and this is, I know you can't read any of these at all, but this is just this is an example of we start breaking things out in more detail, and part of the reason they vary more is because there's a smaller sample size, but it is showing you something that, again, the peatlands rise to the top, but we also have, I think that one is cut off, I think it's calcareous, like beaver wetlands where there's more calcium in the water, or as we're measured through more pH a lot of the time, but tend to score higher. And you see basin shrub swamps scores higher than other types of shrub swamps too, because they're these almost like vernal pools with shrubs in them. And that's something that Heritage is just now starting to create a new classification for. So we'll learn more as they do that. And softwood swamps tend to score higher. Like you think about a red maple swamp, it's this perfect, beautiful, we were in one yesterday, this towering red maple with all these neat plants under them, but especially if you take cover into account, it's the lower score. And I shouldn't. And there's one that says disturbed that's pretty low, and there's also one that says restoration site that's pretty low. And this isn't trying to say that the restoration sites are all in poor condition, but it's also some of those were done at the very start of the restoration. But as we continue monitoring restoration sites, which is one of the things that we're doing, increasing this year, we'll start to see, does the VRAM score go up? And I think one of the really cool things I've seen when starting to look at this is that if a restoration site is near another wetland, restoring the wetland actually causes the VRAM score of the other wetland to go up too, because the VRAM includes looking at the area around the wetland. So we're able to say, look, this restoration is not just benefiting this wetland, it's benefiting other wetlands around it.
Steve has a question. He's wondering if all of those are reference sites.
No.
Okay.
No, and that's something that we'll need more looking at. But, like disturbed wetlands is on there. I don't think we'll ever have a reference site for that. And this is one of the ways our data is messy, is that the natural heritage inventory data, they were specifically going to the best wetland they could find. And, the best floodplain they could find might be more disturbed than the best bog, but still, there's a lot of bias and skew in there we're still kind of parsing out. And on that note, we're not doing complex statistical analysis for the most part yet because our data is messy. We're starting to just look at what correlates with what, and then we're drilling down into it more as time goes on. Because we're looking at as much wetland data as we can get, there's a lot of variability in just the form of the data. Like, we might find a transect instead of a plot and use that data because we're able to generate a number. We track the difference, and I haven't seen that much difference if you average all the transect scores versus all the plot scores, but it's harder it's just another thing to think about when you're looking at the data. And we did have Sean in Watershed Management do some ANOVA, some more complex statistical analysis on a small subset of the data, which was actually really cool because it kind of confirmed, in this case, it confirmed the correlations as having a strong statistical significance, which will come up here. And so here's a look at some of our results we've seen, mostly from those 100 plots. We found that, and this is one that we did do more detailed statistics on, number of non-native species in a plot correlates with poor condition, both in VRAM and coefficient of conservation, even when you take into account the fact that non-natives score lower. Even when you try to parse that out, there's still a very, very strong indicator popping up that not just presence of invasives, but if you have a higher number of invasive species types, the wetland tends to be in worse condition. And then you come into all these chicken and egg things. It's like, well, are the invasives making the wetland in poor condition, or is the wetland in poor condition so the invasives come? And we don't, no ecologist necessarily can parse all of that out yet, but I think both are true. We've found a pretty strong signal also, even in the more detailed statistics, that when a wetland has more disturbance around it, when the buffer is more disturbed and the surrounding landscape is more disturbed, then you'll find more lower conditions in the wetland, even if you're not looking at the VRAM, which takes those into account, but even if you just put a plot in the middle of a wetland, it will tend to statistically correlate with lower condition if the area around the wetland is disturbed. And that's not really a surprise, but it's really helpful to be able to say, hey, look, we have been able to show with this data that if the area around a wetland is cleared, outside the wetland, it will correlate probably with lower condition. We found that this is an interesting one. Doing our water testing, sodium and chloride, which obviously might point to road salts, correlate with poorer wetland condition. But is it that road salt is coming in and just damaging the wetland, or is it that these wetlands with higher road salts have a road close to them, perhaps even going through them, and the road itself is bringing in invasives, is changing how the water flows, etc. So that's something to drill into further, maybe by starting to look at whether or not a road is in a wetland and adding that into the statistics as well. Wetter wetlands are in better condition. Well, what does that mean? Basically, that means that a wetland that is basically, so like you go walk into a wetland, and as you walk in, you see a lot of invasives around the edge. Well, that is wetland, but it's less difficult for a wider range of plants to live in. Invasive species tend to be generalists, so they jump right in there. Buckthorn, reed, canary grass will be more abundant on parts of the wetland that aren't usually saturated or submerged. As you get into the core of a wetland, and there may also be a correlation with edge here we need to look at. Maybe there's more invasives where it's less wet because it's near the edge of the wetland. But even setting that aside, it seems, I've seen this in restoration sites too, where the invasives go away faster deeper into the wetland. And so you get more sedges and rushes and stuff. I actually think if you were to keep going into like a pond on the edge of the wetland or a lake, you might start getting more lower condition again because of aquatic invasives, but I don't have enough data to show that. Habitat diversity tends to correlate with good condition, but again, is it that wetlands in good condition have more habitat diversity, or wetlands with it's chicken and egg always with this stuff. High elevation wetlands tend to be in better condition. I think that's because there's less development and land use around them in a lot of cases. There are also more specialized wetlands and less of these large beaver complexes and such. When humans create wetlands, they tend to score much lower. However, there's the caveat that VRAM is built for natural wetlands, so there may be some issues there with kind of circular logic where, oh yeah, VRAM is scoring a human-constructed wetland lower because we marked that humans have changed the hydrology. So, but we haven't really done much in the way of plots in those. So that's just a very preliminary note. Bog fens and softwood swamps tend to be in good condition, but again, this might be because when they stop being in good condition, they go away. Like if you really mess with the hydrology of a cedar swamp or cut it too many times and the seed bank is gone, it will not be a cedar swamp anymore. It won't be a poor condition cedar swamp. Things we didn't find that one might expect or not. We have not found a strong link between there being a lot of nitrogen and phosphorus in the water and poorer wetland conditions. So, there may be a breaking point at which you dump so much into a wetland that it does harm it, but for the time being, what we're seeing is that, these wetlands have been shown in other studies to filter those things out. And what this might be showing is these wetlands are doing a great job. They're sucking up nitrogen and phosphorus, and up to a point, they're able to do that and continue retaining their high quality. And so that's really encouraging to see, although it's very, preliminary. Turbidity, having a lot of mud in the water, that one's been really inconsistent. Sometimes it seems to link with poor wetland condition. We look at another data set and it goes away. So I think that probably relates to what type of wetland it is. You're not going to see turbidity in a bog or whatever, so we'd probably have to look at a smaller subset of data. Size doesn't strongly correlate with condition. Again, even looking at the plots that don't take size into account, as long as you're in the wetland, a small wetland can have just as good of quality and condition as a large wetland, and it goes to show that they're important and they're valuable, and we want to focus on large wetlands because there's a lot of bang for your buck, but we don't want to forget that the small wetlands are valuable too. Most things like heavy metals and such, a lot of metals we tested for we just didn't find, so who knows how they would affect a wetland, but for the ones we did find, there isn't a strong correlation that we found between having metal in the water and the wetland being harmed. Or being poor condition. In fact, bogs tend to have a lot of iron in them just because of some of the chemical processes in them. So you might even find a positive correlation between iron in the water and wetlands just because bogs exist and it's picking up the bogs. That wetland had a bunch of weird heavy metals in it because it was the lower Connecticut River, and it did have poor condition, but that was one wetland, so it didn't move the whole data. And so I'm probably nearing the end of my time, but I want to talk about, well, what are we going to do next? And then one preview here is you see that Wetlands Reserve Program from NRCS sign. We are working with the NRCS to visit and do the more detailed class 3, tier 3. We used to call them tier 3. Anyway, the more detailed surveys on restoration sites, both before and after restoration occurs, to see how they're responding to the restoration. Because there's a lot of restoration going on, which is really good, but we want to gauge what's happening with it. And we're also continuing to just expand our data set, especially places natural heritage inventory doesn't visit, like they haven't done a lot of work in beaver wetlands. We want to learn more about beaver wetlands. I've started going out in the field doing these level 2 surveys, but there are definitely, well, I wouldn't say it's off to a slow start, but it was off to an interesting start because we were having, this was the day it was 90 degrees, and over in Barton it was like, like early April conditions with this giant flood happening. But this was also really cool because there's a wetland absorbing a flood. Here's a whole pile. So basically when we're looking at places we might go, we collect a bunch of potential sites, and then between asking landowners and access permission and because these are voluntary surveys, a lot of landowners get excited. They're like, oh cool, we want to see what's in our wetland. Some landowners say no or just don't respond, but there's so many wetlands, I'm not worried about it. And we want this to be an outreach for landowners too, so we don't harass. This is the whole pool of sites we identified as maybe wanting the more detailed surveys, and you'll see the 3 watersheds that are colored are the target watersheds for the basin planning process. Most of the other ones you'll see are in Otter Creek or places like that, so those are restoration sites that will either get fall surveys or just VRAM. We'll probably go to other things for our level 2 assessments too, but for the course detailed surveys, those are the main areas we're focusing on, and it looks like we have a pretty good cover of those areas, except for obviously the Vermont Valley to the south. Most of the wetlands are concentrated in the middle of the valley, so there's a lot of points there. Again, as I mentioned, we're doing monitoring to monitor the success of restoration projects all around the state. I guess I already talked about that, and our time is running out, so I'll skip it. But here you'll see we're not just doing monitoring in the restoration project, we're also trying to get plots or at least VRAM and other things. First of all, if there's like you'll see in this air photo, the DESM 1 and 2 are in an intact forested wetland. And this one was a really neat one in that there's like a line on the ground, and the second you got into that forested wetland, it was really good condition, even though it was next to a farm field. So that was really cool to see. So we can compare, because since that's in good condition, that's the goal for the restoration, is for it to resemble that area. So then we're able to look at how those compare, and then over time we'll go back to the site, see if that changes. We're also doing just VRAMs, but we're also looking at wetlands in still farmed areas to get data on what we already know is that a wetland in a hayfield isn't going to be as functional or in as good a condition as in a restoration site. But we want to have that down as data. There's a lot of interest in restoration because on the one hand, there still continues to be development pressure, fragmentation, and also things like climate change that affect everything. And at the same time, there's also an increased interest and awareness of restoration. So we're having more restoration sites going in. But most of the restoration sites are going in large floodplain type ecosystems, which are easier to restore. So, we may not be, if you lose a bog and gain a floodplain, is that really, what you're looking for? So that is, that's going to be a really neat project. Another thing we're doing is developing. We didn't really have a level 1 methodology, so when we're in the office and something comes up, we want to get a feel for this wetlands condition and function. We didn't really have a set way to do that, so we've been working on adapting VRAM to be able to create look at the things we can see, like the size of the wetland, the land use around it. We can see a lot about hydrology now because we have very detailed elevation data from LiDAR, and say, let's estimate VRAM score. And I did that for a bunch of, you'll see most of these are the same points as that other map. So I'm estimating VRAM score for a bunch of wetlands that we're going to go to. And I did that like a month ago. So hopefully I've forgotten, or I'll have Levi do the VRAM for those. And then we'll do a VRAM, an actual VRAM, and see how close the scores are. And my very preliminary result is that it's probably going to be within 5 or 10 points most of the time, unless there's something like the wetland has changed since the air photo was taken. I think it'll be valuable, but we'll see. We'll find out. And this will be really useful for things like if the landowner does say no, we can say, all right, well, are the landowners saying no because they know their wetland's in poor condition and they're like embarrassed or something? So then we can look at this and do this for all of them and say, look, what we're seeing is that we are missing a subset of poorer condition wetlands, especially if we adopt a random sampling methodology in the future. This is another side it's not really a side project. Anytime I draw natural community mapping, I put it on this map. And also when I used to work for Natural Heritage Inventory, I drew some wetland mapping. And so this is this internal notes layer basically that's slowly growing and growing. And we're looking at, well, how can this be useful to other people, or how can we export parts of it to be used? This is not the same as the National Wetlands Inventory map. It's just another layer of, and it's by the same Natural Heritage Inventory methodology. So, and as now that we have full LiDAR coverage of the state, it's becoming really easier to draw these things. So it's been fun to work on that too. And you can see I've done a lot of drawing maps. And so here's what we're looking at moving forward is, like I said, we have kind of interesting data with none of it's bad data, but it's all different. And so in both resolution and methodology, so how do we continue moving forward, analyzing that data to make sure that it's all giving us useful results? How can we, will the tier 1 end up being valuable in that methodology, or do I need to scrap that and go back and try something else? How successful is restoration? We found that it can be pretty successful in floodplains, and other studies have backed that up, but no one has really figured out how to restore a vernal pool, a bog. And if you think about restoring a cedar swamp, well, even if you know how to do it, you're looking at a few hundred years before you're really successful there. So, what restoration sites should we be focusing on, and what wetlands are the most difficult to or impossible to restore? Like with a bog, you might be talking 10,000 years if you get rid of a bog. So, that might have, that has implications. And then over time, we'll be looking at, hopefully we'll pick up signals on like, okay, when the emerald ash borer comes in, how does that affect the wetland? It's not good, but, we'll actually see what happens. Invasive spread, climate change. VRAM doesn't address climate change because I'm not trying to speculate whether an individual storm that dumped sediment in a wetland is climate change or not. But over time, maybe we'll detect a pattern there. Deer populations, I know, are big in other parts of the country, things like that. Unfortunately, we're losing a lot of moose. That probably has an effect. Beavers population, etc. Yes?
Charlie, question from Rose Paul.
Yeah.
Are you seeing any broad trends in deer influence on wetlands dominated by woody species?
That's a really good question, and the answer is right now I'm not, but I think that as we're going down to that Bennington area and doing surveys, this is the type of area where we probably would be picking that up, at least from what I've seen of deer populations. So we don't do animal monitoring right now, and if there's a way, to model where deer populations are higher, I'm sure Fish and Wildlife has data like that. That would be a really, really neat thing to look at. So that's on my pile of like, yeah, I guess that's why it says questions to answer rather than questions I've answered. But
I don't think you just added it to the list.
Yes. But I have heard others speculate that there may be declines in orchids because of the presence of too much deer, trillium, things like that. So it does definitely make a difference. And it's definitely something we'll want to look at. And almost at the end here, and I know it's almost noon, so I'll try to finish quick. So what else are we looking at? We have Levi now, which is super exciting, and he's going to be with us through at least the start of October. So we're going to be able to do even more monitoring than last year. Tina's going to be coming on bioassessment sites, and I'm going to be going to most or all of them. And we'll also have help from some of our other district ecologists and having assorted other people come along for a few of the sites. Bob Zano is going to help us with like at least some tier 2 mapping of some important wetlands on state lands. And we've been talking to the state lands people, making sure that we're monitoring in a way that's valuable to them and meets their needs. Continuing collaboration, we're hoping to connect with more people both through the basin planning process, which has been really successful, and also National Heritage Inventory, the folks over at Fish and Wildlife, both the Heritage Inventory, and maybe I need to start talking more to the deer person too. But all of these, the EPA, we've been, this is funded by the EPA, so we want to make sure that they're, and we would anyway. But communicating with them, just sharing data. I've been in communication with most of the adjacent states at what they're finding, at least roughly. And Brock has actually been doing some connecting with Quebec, so I'm hoping that kind of it moves towards us having a connection with them as well, because they have so many great wetlands and they're so close to the border. Or that's a dumb thing to say. They are the border. and especially when you look at things like climate change, they have some colder habitats. So it's going to be really neat to see what comes out of that. Data and policy, basically just saying, we want to have our policies informed by data, and these types of surveys offer the permitting ecologists the ability to connect back to data when people ask about how the permitting is, when the permitting process has changed, when restoration policies are set, things like that. As natural heritage inventory moves forward, they're doing some really neat conservation planning. We can connect with them. Stratified random sampling is something that comes up a lot, because even the basin planning process is really valuable, but it's not, if we're going to all the wetlands the basin planners are interested in, that might not be a random selection of what the condition of the wetlands are. So what we're going to, but the problem is that you can't select randomly from wetlands if you don't know where wetlands are, and the existing wetland maps are coarse and very skewed towards herbaceous wetlands, so it often just doesn't have forested wetlands. So when it was done in the past, even when trying to correct for that, it seemed like the results were all beaver wetlands. So moving forward, those maps will be improved over time, and I think in the next few years as that happens, we can look at this again. Can we try some random sampling and see if we're actually getting representative random sampling from the data set? And then that will allow us to say for the whole state to actually monitor trends over time more solidly. And then sentinel sites, that means a site you go back to again and again. We've done that kind of informally with places like Bog Brook in the Victory Basin, but if you want to look at things like climate change or changes in deer population, it's really helpful to go back to the same site again and again. And we're also doing that to some extent with the restoration sites, but over time we might look at doing more of that sort of thing. And then in closing, if you have any wetland data that you're interested in sharing, want to see how it fits with ours, if you're interested in learning how to do VRAMs, that's always welcome. I can do trainings, and if you think of other things we should look at, like for instance, I'm not a birder or an insect guy. If you have thoughts on how to integrate those resources into this process, that's all valuable. I was just talking to a wood turtle expert recently about this too. So I've not forgotten about animals, fungi, etc. I just haven't figured out how to add them in yet. So though when a bird flies from one wetland to another, they're not stuck like plants. So they can be more sticky, but they're also important. And on that note, sorry I went over a little from when I went to, but
yeah, so we have time for a question or two, and then if you guys don't mind sticking
that's
around
fine.
a little bit past, if folks have other questions, we can do that. But
yeah, if anyone has to leave, jump in with their question first, I guess.
no, we got to be some typing online.
Okay.
I'll ask a question while we're waiting.
Mm-hmm.
We're about out of time, but I'd really like to have the rivers and wetlands groups get together and really talk about our data.
Yeah, that would be awesome.
Where I've been long interested in the river floodplain wetland connectivity, what the erosion and depositional process emanating from the river affects wetland topography, which then would affect soils and veg. How does channel incision? We know ditching affects wetlands, but how about the actual incision of the river itself and the flood frequencies that we see altered through channel incision? How does that affect wetland function?
Absolutely.
Those are the kinds of things that I'd really like to explore between our two data sets.
That's a really good idea, and I know with the new LiDAR, you're more able to see things like that popping out. And also, I know Levi has done some work. I'm not putting you on the spot, but you've done work in California on incision's effect on wetlands. So I'm excited about seeing his take on some of these sites.
Groundwater.
Exactly, yeah.
And that type of hydrology. These are things that are really fun. We have tons of river data on incisions, so please let's get together.
Yeah, that's a really good idea. Thanks.
I have one question from Rose. Is VRAM data available for download, or at least knowing what sites you assessed?
We don't have it currently in a state that's available for download. Although it would be awesome to, we do plan to at some point, but I can definitely send either an ARC layer or a Google Earth layer or something of all the sites that we have, and I'll just make sure that I connect with her later.
Yeah, Rose, email Charlie.
Yes, send me an
email. A big goal of ours.
Yes.
Is to have this all publicly available to interested folks.
Yeah, exactly.
And to have them also, input data, them using VRAM and inputting their data into like some sort of like web portal for us to look at as well.
Exactly, because we don't want to say, hey, Nature Conservancy, give us a bunch of data, throw it into this hole, and you'll never see it again. We want it to be so that, you add data and you also see how it compares with the other data that other people have gathered. So, yes, it will happen, and in the meantime, send me an email.
Well, we're out of time, but if anybody wants to stick around, Charlie and Tina can answer more questions. I'm going to turn off the recording, but again, thank you to both for presenting today.
Thanks for inviting us, Bethany.
Sure.
Yeah.
And our next lecture is scheduled for the 14th of June. And stay tuned for the topic to be confirmed. And that'll be wrap up the 2017-2018 season that will start again in September. New series. So thank you.
Thanks. Stand up and stretch.
Feels good.
Yeah. Sign ourselves in.